Thank you very much. Good afternoon. My name is Peter Sabel, and I'm a postdoc in Christian Funkhauser's lab. And I'd like to introduce my poster, which is about a small family of proteins named PKS from phytochrom kinase substrate, but they actually also work in phototropin pathway, and that's what I'm interested in. And I've tried to elucidate the function of those PKS genes by looking at auxin-related processes and with collaboration with Matthias Fendrich lab also by looking at pH changes, or at least preliminarily at pH changes in the cell wall during phototropism. This is not actually how my poster looks like. It was before I had finished the poster, but so if you're really interested how my poster looks like, I invite you to visit poster number 2. Thank you very much.
Hi, my name is Nikhil Job. I'm a postdoc in Professor Liam Dolan's lab at GMI Vienna. I'm investigating how a meristem is formed de novo in Mercantia sporelings. So we are using Mercantia spores as systems to study how a meristem is formed from a non-meristematic cell. And precisely, I'm investigating how light is modulating this process. And my analysis shows that red light rather inhibit and blue light promote the formation of a particular developmental structures that forms meristem in future. So currently I'm investigating the molecular mechanism behind this observation, and please stop by my poster if you would like to know more about it. Thank you.
Hello, everyone. My name is Hanhong Liu from the Institute of Botany, Chinese Academy of Sciences. Today, I'll briefly introduce my research on retrograde signaling during photomorphogenesis. So upon dark to light transition, a harmful molecule called singlet oxygen is generated and perceived by executor proteins. Then these proteins translocate into the nucleus and alter gene expression. However, the underlying mechanism of how X proteins enter the nucleus remains unclear. So our research question is how do executed proteins enter the nucleus during retrograde signaling? So our results show that X proteins interact with multiple nucleoporins of the nuclear pole complex, and mutations of those nucleoporins would inhibits the nuclear localization of X proteins, as well as mitigate the singlet oxygen response. So for more information, please come to my poster number 6. Thank you.
Okay, Stefan. Thank you.
Thank you. I have a poster here. I've not actually seen it yet. It's been kindly made for me. So we're going to unroll it shortly, and it's going to be pretty much on the things that I talked about earlier.
Hello, I'm Kotaro Akita from Yamagata University. It is well known that auxin plays pivotal role for tropism. However, its role in hydrotropism of Arabidopsis was not elusive. So we investigated the role of auxin biosynthesis, transport, and responses of hydrotropism of Arabidopsis. These are the typical results, and we found that these processes are necessary for attenuating hydrotropic bending of Arabidopsis. To combine with the result of MIS1 and MIS2 mutants, we demonstrated that auxin acts downstream of MIS1 to attenuate hydrotropic bending to fine-tune the root growth direction. For detail, please visit our poster. Thank you.
As we've heard over the last days a couple of times, auxin is the most important hormone in plant tropism. But without transport, auxin would just be a molecule. So this makes the PIN proteins, which are essential in auxin transport, the most important transporters in plant tropism. To better understand how these transporters work, especially regarding autoinhibition on a mechanical level, as well as the interaction with activating kinases, I took a bashing approach. By truncating the loop domain of PIN3, I aimed to find specific interaction sites as well as sites involved in autoinhibition. To furthermore identify sites involved in interaction between the different domains, the loop domain and the transmembrane domain, I played a bit of Lego with my pins. And if you're now curious to find out how playing around led to the discovery of two crucial loop domains regarding autoinhibition and kinase interaction, as well as new insights into domain interplay, then please come find me and the hard data at poster number 12.
Hello, everyone. My name is Katarina Kurtovic, and I'm from Jan Petrasek Group. And today I would like to present you something completely different. It's a streptophytic alga called Hara brownie, which is a close relative to land plants. And I'm truly honored to be presenting it here today in Japan, since Japan has such a long history of Hara research. And even whole genome sequencing was done on Japanese strain. And in my poster, you can see two side projects because one is simply not enough. And I was accidentally finding this first discovery by time-lapsing some Hara growth. And I could see the tip branchlets at the tip are opening and closing in circadian rhythm. I tried to search the literature, but I couldn't find any mention of it. So if you would give me your thoughts or comments to it, I would greatly appreciate it. And the second project relates to phosphoproteomic analysis of Hara under after auxin treatment, where we found some really exciting new candidates that might be involved in auxin signaling. And if you're curious to know more about these new cool movements or finding about these candidates or even my main project, which are Hara pins, make sure you check out number 14. Thank you.
Hello, everyone. My name is Amir Ohad. I'm from Tel Aviv University at Dr. Yasmin Moroz's lab. And my work is about twining initiation in climbing bean plants. So what we do is I actually look at impacting circumrotating bean plants onto a pendulum-like support, and we can measure the deflection of the support off of the vertical. Using this deflection angle, we can measure the force applied by the beans onto the support, and we can compare these force trajectories with numerical simulations, which we find to match very well. However, we do find some mismatching when the shoots deflect off of the support, and the angle we would expect from the circumnutation if it was not impeded by the support is smaller than what we see. And so this is determined by the amount of contact time you can see with the linear correspondence on the right. So if you want to see more, you can come to my poster number 16, and I'll also be presenting this in more detail tomorrow morning. Thank you.
Hi, I'm Kazuo Ebine from NIBP, and we are analyzing the molecular mechanism of membrane trafficking system in plant cells. And pollen tube guidance is one of the important chemotropic response in plant cells, and PLK6 acts as a receptor of chemoattractant LUA1 peptide. And it is reported that when LUA1 is treated, PLK6 accumulates on the plasma membrane specifically to the LUA1 positive side. So maybe LUA1 interaction inhibits endocytosis of PLK6. To understand this mechanism, we focus on the adapter protein clustering-mediated endocytosis. And recently, we found that PICAM2 mutant shows a defect in the LUA1 response in wavy assay. So now we think that PICAM2 regulates the endocytosis of PLK6 specifically to the LUA1-mediated systems. Thank you very much.
Hello, everyone. My name is Minoru Noguchi from Utsunomiya University, Japan. As you know, a blue light receptor phototropin induces various blue light responses. And in previous study, we found that photo2 is a thermosensor in chloroplast movement. However, the involvement of photo2 as a thermosensor in other blue light responses is unknown. In this study, we demonstrated that the cold treatment in darkness promote subsequent blue light responses such as stomatal opening and phototropism. And mutant analysis showed that this response is depending on photo2. So we named this response photo2 mediated daily cold priming. And in the poster session, I'd like to discuss the pathway and biological significance. Poster number is 20. Thank you.
Hello, everyone. So my proposal is about our recent observation in the rice root, how it responds to ammonium nitrogen. So it's actually a brief description of my talk in the morning. So if you have questions, please come to me.
Hello, my name is Tatsaki Go from Nara Institute of Science Technology. I talked yesterday. So we root can change the growth direction in response to gravity. To know, in order to know how change the growth direction, we try to time-lapse imaging and found that the changing happened by phasic way. So I'd like happy to have a discussion with you. Thank you.
Hello, I'm Hiromasa from NIBB. We are studying gravity sensing in plants. And as you know, the priority of the lazy protein on the plasma membrane is established by the translocation from the sedimented aminoplast onto the nearby plasma membrane domain. However, we have a little about how the protein is maintained or erased. So we focus on the protein stability of the lazy on the plasma membrane and found ubiquitination of the lazy is one of the mechanisms to regulate the stability. Thank you.
Hi everyone, I'm Ronnie. I'm from Tel Aviv as well, actually from newly Professor Moroz's lab. And we are so we heard a lot about all sorts of external signals affecting plants. And we heard that Malcolm is very bothered with the clean 90 degrees angles. And in the context of light, we also said, we keep doing these experiments with light only from one side. Maybe it's interesting to look at something a bit more natural, or right, in nature you have things a bit more complex. So we built this setup that allows us to take a first step and just use 2 light sources. And then we collect the trajectory of the movement of the plants that we subject to these different light conditions. And this allowed us to corroborate a mathematical model where we can predict the movement of the plants, the growth trajectory. Yeah. And then we also have some surprising results for what happened when we added a 3rd light source. And if you want to hear more, you'll have to come to the poster. Thank you.
Good afternoon, everyone. I'm Chung Uri from Gyeongsang National University. Today, I'm going to talk briefly regarding my poster, oxygen signaling repressor IA8 promotes seed germination by downregulation of ABI3 gene in Arabidosis. The key question is, how does oxygen signaling regulate seed germination under stress condition? So we have found 3 evidence. Firstly, seed germination is reduced by IA8 mutant under stress condition. Secondly, IA8 inhibit oxygen signaling in response to stresses. Finally, IA8 promotes seed germination by the secretional inhibition of ABI3. In conclusion, IA8 acts as a positive regulator during seed germination. And inhibition of oxygen signaling by IA8 is crucial for cell germination under stress condition. Thank you for your attention.
Hi, I'm Yoshiro Murakami, first year PhD student from Kyoto University. This is Arabidopsis glycasin. This gene is called two different type protein, not by alternative splicing, but alternative TSS or alternative promoter selection. Longer mRNA called plasmid localized glycoprotein and shorter mRNA called cytosol localized glycoprotein. And recently I found only shorter mRNA is unstable in cytosol. And the stability of mRNA is changed by light condition. So to be continued to my poster number 32. Thank you.
Hello, my name is Mike. With my poster, I am introducing a new sensor line for auxin, and it's based on the autonomous fungal bioluminescence pathway. It has a constitutive biosynthesis of luciferin and the DR5 expression for luciferase. The line is stable and usable by auxin, and auxin sensitive without even auxin. And it looks like here in this photo. You can take photos of this with simple cameras or with also more elaborated cameras. Both for my sensor and me, this is a new part of experiments for tropic responses. And I tried two different type of experiments, one with red light and blue light, and one for STEM gravitropic and phototropic response. And I think that I have seen some differences there with red light, and particularly in the leaves of the rosette. And for the STEM gravitropic and phototropic response, I could see some induction of auxin, mediated auxin-driven bioluminescent in the lower part of the stem. In the lab, I'm interested to expand these experiments with applying more auxin inhibitors and involving also mutants. Also, I'm working with roots in rhizotones and plants growing in normal in soil. You can see down there at the right. And also, we have a number of other sensors that is for other hormones. You are all invited for any kind of interactions in the poster 34. Thank you very much.
Hello, I'm Kaisen Maruyama, a PhD student from the Laboratory of Plant Regeneration and Morphogenesis in NICE, Japan. So literally, my topic is about plant regeneration, especially like de novo shoot formation from cows. So my poster has two parts. So one is about WOX13, which is a kind of Wuxia-related homeobox transcription factor. And our research group recently found that the role of WOX13 in shoot regeneration, and that WOX13 represses a shoot formation on shoot inducing media, SIM. So although the WOX13 is already expressed on CIM, so before being transferred to SIM, the function of WOX13 on CIM is still elusive. So we are looking for the WOX13 downstream target on CIM. And also, I'm so interested in the auxin, like auxin biosynthesis or like degradation of metabolism. So I like to discuss the auxin, the significance of auxin biosynthesis in the shoot regeneration. So my topic isn't about tropism actually, but I'd be really glad to see you in front of my poster number 36. Thank you.
Hello, everyone. So I'm Akihito Mamiya from the Fukaki Lab. And in poster 38, we are presenting our work on root-shoot differentiation. So what we did is grab a bunch of lateral root mutants and put them through the shoot regeneration two-step protocol, in which we induce callus from seedlings and then regenerate shoots from them just for fun. But what we found is that actually the auxin-inducible LBD transcription factors that are redundantly required for lateral root development show an interesting phenotype. So first, in the callus, well, the mutant callus has lost the dome-shaped lateral root primordium-looking structures observed in the wild type. And second, the mutant callus regenerated way more shoots than the wild type. In addition, the mutant callus had increased expression of root tropism-related genes, including ADF9, STR9, MISCUS1. And so if you're interested in what the hell tropism has to do with this, please visit my poster at 38.
Hello, everyone. I'm glad to introduce about our research work. So we are interested in molecular mechanism of apical development. So MUR1 is an enzyme which makes GDP-fucose, which is required for xyloglucan and pectin RG2. MUR1 mutant is defective in RG2 dimerization, and interestingly, boron rescued it. Hook is formed when the cells on the outer sides elongate faster and the cells on the inner side elongate slowly. And we tried to identify the molecular mechanism, how this boron rescued, and please visit for more discussion to my poster number 40. Thank you very much.
Hello, I'm Denisa Ulehlova from Charles University in Prague. And in the group of Mateusz Fendrich, we largely focus on rapid auxin signaling, which is crucial, for example, for initial stages of gravitropic events. And in the search for some novel players in this rapid signaling, we bumped into a fosducin through the phosphoproteomic screen. And this particular fosducin-like 2 protein, it was shown to interact with the receptor of heterotrimeric G proteins, RGS1, but besides that, nothing is known about this guy. So we show that this is essential gene, its knockout is lethal, but the knockdown shows some very interesting auxin-related phenotypes, such as these excessive waving of primary root, this apparent agravitropism, crooked hypocotyls, problems with maintenance of apical hook and such things. Yet what we show that these plants actually do sense gravity, they do respond to it. And we propose that this is like basically problem with the maintenance of direction rather than the gravitropism problem itself. If you want to know more about this novel, interesting guy, please visit me at poster number 42.
Hi, everyone. My name is Huilun Sun. I'm from Institute of Botany, Chinese Academy of Sciences. My research investigates how PAF4 and its ubiquitin ligase regulates the degradation of phytochrome B. Phytochrome B is a photoreceptor sensing red and infrared light in plants. And in previous studies, PAF4 is a multifunctional protein. It can coordinate many pathways in plant growth, development, and environmental responses. But so far, it has no functions in light signaling pathway. And here we found PAF4 has negatively regulates photomorphogenesis under red light with shorter hypocotyls in PAF4 mutants. And we further give evidence through protein interaction, protein level regulation, and modification to prove the new function of PAF4 in light signaling. And welcome to my poster at number 44. I'm happy to discuss my research with you. Thank you.
Good day, everyone. My name is Rian Maliwat from NICE, and my research mainly showcases the functional connection between PRRs, which are clock genes of the plant circadian system, and phototropins, which are blue light receptors that regulate photosynthesis. So compared to the phytochromes and cryptochromes, little is actually known about the relationship between the circadian clock and the phototropin receptors. However, with previous reports stating that the PRR clock genes are significantly expressed within the mesophyll cells and that they can regulate sucrose synthesis, we are highly convinced that these clock genes are somehow involved in the phototropin signal transduction pathway. So to verify these assumptions, we analyzed chloroplast movement in PRR misexpressing lines, and we were able to observe that this particular activity is strongly influenced by the PRRs. In addition, we were also able to observe that the accumulation response may be working in a time-dependent manner. So if you want to know more about the details of this newly found connection between PRRs or circadian clock and phototropin signaling, please visit poster number 4 to 6 later during the poster session. Thank you.
Okay. Good afternoon, everyone. My name is Woo-Sik Jung from Gyeongsang National University. It is well known that hysteresis inhibit flower development. However, the detailed molecular mechanism was not identified yet. So our question was how flower development is inhibited by the hysteresis. We found several things. First, our finding was IA8 is stabilized by the phosphorylation of the MAP kinase in response to the hysteresis. Second finding was IA8 phosphomimicking overexpressing plant show defect in flower development. And the third one is expression of the flower development regulated genes are disturbed in the IA overexpressing transgenic plant. I will be happy if you have chance to discuss more detail in the poster. Okay, thank you.
So, okay, thank you everyone. So, shall we start today's posters in conference room B? And after the poster session, as scheduled, we will start session 8, 4:15 p.m. So please come back here again. Okay.